Welcome to Chemistry 1, Unit 1 Matter, Lesson 4, Video 2. So in this video, we are going to look at separation techniques, ways that we can separate mixtures, and then we're going to see some demonstrations of this in lab. So our first one is evaporation. Evaporation is just the removal of a liquid from a mixture. More than likely, it could be water. So if you let salt water sit out over time, the water can evaporate and we're left with salt. That's how we get sea salt to use for cooking. If we want to speed up the process, we can add heat. So heat speeds up this process. And we use this for suspensions and solutions. So this is a possibility to use to separate suspensions and solutions. Filtration is one that we saw in our first lab. So here, solids are separated from a suspension. So you might have a cloudy mixture. The filter paper actually catches the solid, the very small solid particles, and allows the filtrant to come through, and we end up with a clear solution. A centrifuge. I'll demonstrate one of these in class. They're pretty neat. But this is a spinning force. That separates small particles from a liquid. So this can be used for suspensions. Hospitals use a centrifuge to spin down blood. So blood is in those little tubes when you go have blood drawn. They can put that tube in a machine that spins it around really quickly, and the blood cells actually go to the bottom of the tube and you're left with a liquid plasma. That plasma, the liquid part of blood, is kind of like a straw colored, light yellow, but it's clear. So we can separate other things. So if we have muddy water, we could spin it and we could get the dirt particles to settle at the bottom of the tube. So centrifuges are very common in biology labs. Decanting is something that we know already because we went over this the second week of school. So this is the pouring off of a liquid from a settled solid. So this is also used for suspensions, but only ones that will settle over time. Magnetism, I really don't have to explain, but this is used to separate substances that contain iron and some other metals like cobalt that are magnetic. So with that, you can even separate very small particles of iron. Total cereal has little tiny wee particles of iron added into it. It's needed by our body, and it's so small that we can't detect it when we chew. But if we were to crush up total cereal and we were to put it in water and we were to put a magnet in there, we would actually see little tiny wee spots of black that are attracted to the magnet that are little wee iron particles that they add. It's a very, very fine powder that just mixes in with the cereal when they make it. Chromatography is something that we'll demonstrate in class. This is the use of a solvent to separate pigments or other substances based on size. So you might have done this in biology class where you separate out the pigments that make up chlorophyll, the green pigment in plants. There's actually 4 different pigments in there, and they travel up through a piece of paper. The big pieces can't travel as far as the small pieces, so one pigment goes higher than the other. We can demonstrate this with different colors of marker to see what they are made of, what different colors. So like a black Sharpie is going to have red, yellow, and blue pigments in it mixed together to give us black. And then distillation is the last one. This is used to separate mixtures of liquids and solutions. And it uses differences in boiling points. So distillation is used in the manufacturing of spirits and alcohol. That is because the ethanol that's made by the yeast bacteria has a lower boiling point than water. So if you take that mixture that has ethanol or alcohol and water and you heat it between the boiling point of the 2 substances, one of them is going to rise up and you can collect that gas and leave the other one behind. So with making a vodka or any of those substances, the alcohol boils and we collect the vapor and we cool it down and it condenses back into a liquid. We're left with water left behind. So that process is tricky because you could also make some very poisonous and toxic substances if you don't know what you are doing in that process. So those are all different separation techniques, and we will demonstrate some of them in class.